Thanks a lot for the opportunity to be here and explain a bit what I'm doing and what we are setting up here in Bonn. And yeah, why this topic on greenhouse gas measurements is relevant, and also because I feel there might be opportunities also for collaboration or for potential joint projects here with CEF. So yeah, I'm going to talk about how greenhouse gas measurements can help us develop climate smart agroecosystems. But for that, we are first going to briefly discuss or present why greenhouse gas measurements are relevant. And I mean, everybody knows climate change is happening and that the concentration of these gases in the atmosphere is increasing. So here I present some data from the IPCC, carbon dioxide, but also methane and nitrous oxide. So we talk very often about carbon dioxide, but in agricultural systems, these other two gases, as you may know, are very relevant. And then we see how, since the beginning of the Industrial Revolution, the concentrations in the atmosphere of these three gases has increased exponentially. So if we take here a zoom at this period from 1960, we see that CO2 was about 320 ppm, and now it's about, in 2020, 409. And then we see here that the increase is not constant, that there is an up and down every year, a peak up and down. And this is because the Northern Hemisphere has a lot of green covered land, so forest and also croplands, that during the spring and the summer, they take up a lot of CO2 from the atmosphere. And this can generate the little dents even in the concentration worldwide. For methane and nitrous oxide, we don't see these variations so clearly. There's some patterns, but we also see that these concentrations are increasing compared to previous values. And here, these data are already from actual measurements in the atmosphere, while previous data here are from ice cores that allow us to get this information. So we see that this concentration, for example, in the period 2021-22, has been increasing for the three gases, and of course at different rates. But it's important to look at this table to see that we talk very often about CO2, but methane has a global warming potential, so a capacity of warming the atmosphere 27 times higher than one molecule of CO2. And this is because of the capacity of taking heat, but also because of the lifetime that it stays in the atmosphere. And the same for nitrous oxide, the value is much larger. And therefore, even if these emissions are much smaller, it is very relevant to also look at these two other gases. So where are these gases produced? And this is global greenhouse gas emissions without splitting into the different gases. So energy takes a very large share. And we are going to be looking today at this part, AFOLU, which stands for agricultural, forestry, and other land uses, and which is responsible for about 22% of the global emissions. And then in agricultural systems, where is this happening? So here is agriculture and enteric fermentation. So this is methane produced by cattle. Is responsible for a large share. Manure management, and then nitrous oxide from agricultural soils. And today we will talk a lot about nitrous oxide from agricultural soils and maybe some of the other sources, but also croplands here for CO2. How can they affect these emissions? And here we see that forests are a large carbon sink. So this is also something we should try to keep. So agriculture is responsible for 12% of the emissions in Europe and 7.4% in Germany. This is not because German agriculture is so well managed that the emissions are very low. It is because the share of industrial emissions is very high. So then this makes the number from agriculture or the proportion of emissions coming from agriculture be a bit smaller. But still, the net amount is very large, and therefore there's opportunities for climate change mitigation. But before we decide on how we can implement mitigation strategies, we need to be sure, okay, how much are we emitting and what is the effect of the different practices that we may do. So here we are looking at the AFOLU sector, so the agricultural, forest, and other land uses. And exactly, so we have here the synthetic fertilizer application with nitrous oxide, which is only a 3%, manure management, rice cultivation, and this is also something we will talk about today, which also plays a relevant role, and managed soils. You can see here the share of emissions from the different sections. And if we focus on Europe, not all the data we'll present today are from Europe, but mostly we see that there is not large emissions from land use and land use change, the green one, which globally are still very relevant because they are very relevant in Latin America or Africa or Southeast Asia. This is because land use change, deforestation is still happening at a very large rates in these regions, and this is why this is very relevant, while in Europe, there's not so much land use change happening, and forests are capturing large amounts of carbon, or certain, important amounts of carbon. And then for Europe, enteric fermentation is very relevant, and then managed soils and manure and synthetic fertilizer. But soils or agricultural soils can also store large amounts of carbon. There's more carbon in the soil than in the vegetation and the atmosphere together. In fact, there's some studies showing that the saturation on carbon in managed soils is much lower than in natural soils. So then there is an opportunity to increase the carbon saturation in soils so that we can store more carbon. And for this, we're gonna look at different strategies or management practices that make us understand how can we increase the carbon in the soil without modifying the emissions. Or without having a negative trade-off with the greenhouse gas emissions. So if we see here, this is an example of a natural or of a cropland where we are having emissions from the 3 most relevant greenhouse gases, and through different mechanisms. This is very simplified because there's, for example, for nitrous oxide, many more processes taking place. But there are sources and sinks of these gases, and the soil can take up a lot of carbon. So what we need to understand is how can we make the soil take more carbon without leading to increasing emissions. So what we do in our group, we try to aim at climate smart agriculture practices. And climate smart, the FAO defines it by management or a type of agriculture that sustainably increases productivity and incomes. It's adapting to climate change and mitigates greenhouse gas emissions. And what we are going to focus today is in this section here. I'm pretty sure many of you work a lot in the sustainable increase of productivity and incomes, and even in adaptation. So what we are doing is a bit of adaptation, but also mostly mitigation. But to develop these mitigation strategies, we first need to quantify the gases, the emissions that happen under different soils, under different climates, and under different management strategies. Otherwise, we cannot say this management practice is good for mitigation or not if we have not tested them. And for that, we use different methods. And I would explain a bit how these methods work so that you understand also how we measure in the field. And these are the most common methods, so chambers, and some of you might know them, and eddy covariance flux towers. So chambers are just kind of a bucket that you would close on the soil and plant system, but you could also do it on the soil, and for a certain period of time. And during this period of time, we would regularly take gas samples and analyze the concentration of the gas we are studying every certain period of time. So here it is every 10 minutes. We measured CO2 and at 4 points. And then we would calculate the linear increase in concentration with time within the chamber's headspace, and we will estimate the fluxes. This is how it was usually done. luckily now we have analyzers that measure at much higher frequency. So instead of having closing for 40 minutes and having 4 points, now we have analyzers that measure every second. So we can close for 3 minutes and have lots of points. So this is an example of one of our trials that I will explain later, where we just close the chamber and increases the concentration of nitrous oxide. We open the chamber, it goes down, we close it in a different place, increases up. And then using exactly the same method as we did before, we can estimate the flux by calculating the ratio of increase in concentration with time in the headspace. So these chambers have the advantage that they are quite easy to manipulate, and you can move them around and measure greenhouse gases in different plots or under different conditions. And they are generally not very expensive, so just time consuming, and produce data only from a small surface. So then the alternative is using these eddy covariance flux towers that basically work as if we would have an imaginary large chamber that we close over our ecosystem. And then we have, everything went, an imaginary flux tower, we close our chamber, so an imaginary chamber, our flux tower in the upwind direction, and we are trying to estimate the exchange of fluxes over this imaginary flux tower. There's a lot of physics and assumptions behind, but the idea is that on top of the tower, we have a gas analyzer, an anemometer. This is a sonic anemometer that measures the wind direction very, very frequently. So this is 20 times per second. And then the concentration of the gas we are interested on. In this case, this is an analyzer for CO2 and water. And with that, we can estimate the fluxes within this imaginary chamber. So this allows us to get a flux calculator for a much larger surface where these exchanges are taking place. And then just as a convention in the field, we assume that when the fluxes, the ecosystem, so soil plus plant system, is emitting to the atmosphere, we consider the fluxes positive and they are a source to the atmosphere. And when the ecosystem is capturing whatever gas we are looking at, it's negative, and then this is a sink. Okay, so this is the convention in the field. So now I'm going to present some of the studies that I've been involved in, from rice paddy fields in Italy to olive groves in the Mediterranean areas, oil palm plantations in Indonesia, and end up with some of the case studies the trials that we have now in Germany. So this is a rice paddy field in Italy in the Po Valley. So this is one of the largest regions producing rice in Europe. And then the rice is a large methane production, a crop that produces a lot of methane, as you may know. So what we did here is we installed a flux tower and also some chambers to measure methane, CO2, and nitrous oxide, so the three gases. At that time, it was not possible to measure nitrous oxide with the eddy covariance, so this is only with chamber data. For the rest of the fluxes, we have both techniques. And we measured during 2 full crop seasons. And this is CO2, and you could look just at the blue dots, which is how the ecosystem is capturing CO2. So we started in January, and the emissions were close to 0. Then here the rice is sown, and then rice starts capturing a lot of CO2 until it reaches the peak of maturity, and then starts drying out. And when it's fully dry and after harvest, the emissions go back to 0 again. And not much happens. There are small peaks here and there during the intercrop periods. This is very ecosystem specific, but here it's very cold in winter, even if the summer is very warm. And then next cropping season comes in the following year, rice is harvested again. Here there was something happening, I will explain in a minute, and then goes back to 0. So in these periods, large amounts of carbon are captured. Not much is happening here. These other 2 lines is just the partitioning between this net ecosystem exchange, which is what we measure between ecosystem respiration and photosynthesis. So here we have the methane fluxes, and we measure with eddy covariance. So these continuous measurements are the gray points, and the black points are the more spontaneous chamber measurements. And even if we are missing a lot of periods, this was kind of the first measurements ever with methane eddy covariance on a long term. So it was quite tricky to keep this running, and that's why there are some gaps. But still, and we were also measuring with chambers. The chambers, as you see, don't have the same temporal resolution, but we see that the emissions go up and down. And then the second year, what we did is we implemented a mid-season drainage of the water table, which is one of the management practices which is well known to decrease methane emissions. And we wanted to test if this was really the case. So we decreased the water table for a couple of days, and then we reflooded again. And we were measuring in parallel nitrous oxide, because one of the trade-offs of this management practice is that it does usually decrease methane, but it has the potential to increase nitrous oxide. And as we saw at the beginning, the nitrous oxide has a global warming potential nearly 300 times larger than methane, than CO2. Then the trade-offs can be interesting. So here we try to make a balance of all the gases, and I'm not going to go in detail because there's quite a lot, but what we wanted to see is that the methane emissions from one year to the second one, they were clearly much larger on the first year, while the nitrous oxide emissions were a bit higher on the second year, but not so crazy large. So when you make the global warming potential, which accounts for the CO2, methane, and nitrous oxide, the year where the water table was kept continuously, the emissions were much larger. But of course, this would be if we would only look at the emissions. What happens when we take into account also the yield? And this is what, in climate smart agriculture, yields are important. And for farmers, you need to also consider the yields. Otherwise, the proposed management practices do not stand. So then the yields were also much lower on the second year when we removed the water table. So because keeping the water table has the advantage of preventing the weeds to develop. So if you remove it, you let it develop, there's some things that, the yield was much, much lower. So then is why we set up this unit, which is the greenhouse gas intensity, which relates the greenhouse gases with the yield as the only way to kind of check for the trade-offs between both things. And still, the greenhouse gas intensity was much higher. But yeah, before you could offer this kind of management practice, it was suggested you would also have to consider the trade-offs with yield. Okay, and now I'm going to jump to a different ecosystem, which is an olive grove in Spain, where we were comparing the difference between keeping the weed cover between the olive trees or the traditional management practice, which is removing everything with herbicides, because they are worried about the competition with water for the olives, the competition for water. So we measure also with two flux towers. So we had here two paired systems with two flux towers where we measure with the eddy covariance technique CO2 fluxes. And this is 1 year only. And we see that the weed cover captures way more CO2 because, of course, the green grasses that are growing between the trees are also capturing CO2. So this, in terms of annual net ecosystem exchange, is much more positive because it captures more CO2. Then we wanted to check, okay, what are the trade-offs with other greenhouse gases? Do we see any difference with other greenhouse gases? And we saw that there is no difference in nitrous oxide emissions, and there was a slightly larger uptake in the weed-free treatment, so the methane emissions were a bit more negative. But when you make the balance with all of them, the increased carbon capture with cover does not result in a trade-off, a negative trade-off with the increased greenhouse gas emissions. So this could be something that we could look at. Again, here, I'm not showing the results, but also the yields were a bit lower. And there's theoretically, now that there is some irrigated olive groves, that could be a solution. But of course, if the yields are also decreasing, then this also gets complicated. And we jump now from an olive oil plantation to an oil palm plantation. And this is in Indonesia, where we also measure ecosystem fluxes using the same techniques, flux towers and chambers. And here what we compared was a young oil palm plantation at very early stages of development, where the palms are not producing any yield yet, so they are just growing but not producing yield, with a mature plantation, which is already productive, 12 years old, which was already producing a lot of yield. And then what we can see here is that the young plantation was a very strong source, and the mature plantation was a very strong sink. So in these 12 years of development, the plantation moves from being a very large source to a very strong sink. So then we also looked at the nitrous oxide emissions and the methane emissions. We are working here now on mineral soils only. So we measure carbon and nitrogen in the soil. Soils are mineral. We measure nitrous oxide, and the emissions in the mature plantations were more or less the N2 emissions 3 times larger. This is because they are very heavily fertilized. So then the N2 emissions are very large, and methane emissions are basically the same. So we think here that they are very heavily fertilized, but also that here we were measuring an industrial plantation, where the fertilization is larger. So the effect of smallholders versus industrial plantations plays here a very relevant role. And then in this second part, we looked at peat soils. And peat soils have very large carbon contents, very large nitrogen contents compared to the mineral soils, both mature. And what we see here is that the CO2 that in the mineral soils was a strong sink, in the peat soils is no longer a sink, but it's also a strong source. So it is very important not only to take into consideration the development stage of the plantation, but also in which type of soils this is growing. And also for the nitrous oxide emissions, in the peat, they were much higher. And in case of methane, the system stopped being a sink to become a source. So then we took, so this is the full overview of the whole life cycle of the plantation. So then we tried to do life cycle analysis for first rotation and second rotation cycles of plantations. And in this particular case, we did it for palm oil biodiesel. So trying to understand, because at that time there was still the discussion if palm oil biodiesel was something we should support or not. And we kind of challenged the traditional assumption of life cycle analysis that for croplands, they assume that whatever the crop captures is later emitted and is not estimated. And what we did is take the real field measurement to update these numbers, and using the real numbers to make an improved life cycle analysis, where we saw that, so this is the traditional analysis, and we made an improved life cycle analysis where we took into consideration all emissions up to the cultivation of the palm oil. When we made the analysis for palm oil biodiesel, and we saw that this was very negative. And then we are trying to set up, okay, we've looked at specific plantations, but what is the footprint of all the plantations? So as we've seen that it is very relevant if we are looking at mineral or peat soils, if it's early or young, if it's smallholder or industrial plantations, but also first or second rotation cycles. So here we are trying to take data, and here I'm only presenting data from Indonesia, Malaysia, which are the largest palm oil producers in the world, but we are expanding this analysis to global cultivation, considering FAO data from plantations, how the soils were converted, looking at land use change maps, but also satellite images, and understanding what the emissions are at different stages of plantation. You can make analysis of what the emissions at a certain point are. No? And then what we are trying to see here is, okay, this is what's happening now, the current emissions that we have, and how can we have mitigation strategies? How would we develop some opportunities to, okay, the emissions that are done right now are done already. There's nothing we can do. But in the future, how can we propose strategies to reduce the emissions? So as most of the emissions in our analysis, we see that are caused by plantations on peat and from forest conversion. So first rotation cycles and plantations in peat. So our mitigation scenarios and the yellow, this is the percentage on mitigation protection. So this is mitigation via land protection, so not using forests, through intensification, closing yield gaps, and then the minimum historic baseline that is not possible to solve it because this is already, so this would be in 2050. And then we see that if we stop using new land for plantations, if we stop using peat soils and if we start converting forests, then there's opportunities for decreasing a very large amount of the emissions, up to 40% of the emissions in 2050. Okay, and then in this jump to Asia, we're going to go back to Germany, and I'm going to talk about one management practice that is reduced tillage, which is very often proposed as a strategy to increase carbon in the soil. So this is typically suggested as the way to go to reduce tillage or no tillage as management practices. And there's several long-term trials all over Europe to test if this is happening. And here we are using a trial in Göttingen where it was set up in the 70s to compare long-term, on the longer run, conventional tillage with reduced tillage and see what are the effects. And they were basically mostly looking at yields. So these are data from an analysis done just a couple of weeks ago, where we collected soils up to 60 centimeters, because previously we had only looked at the topsoil. And if we looked at the topsoil in the reduced tillage, indeed, the carbon content is much larger than in the conventional. So we would say, okay, yes, this is a good strategy to increase carbon in the soil. However, when we started doing our analysis up to deeper layers, because the reduced tillage only goes to up to 7 centimeters, then indeed, yes, you do get a bit higher here in the topsoil, but at deeper soil layers, the carbon never reaches. So then in 20 to 30 centimeters, in the conventional tillage, the carbon content is much larger. And this is after 50 years of implementation of this management practice. So when we do look at the total carbon stock of the two tillage systems, we see that there is no difference between conventional and reduced tillage. So in this particular place, with the type of soil, the climatic conditions, after 50 years, we see no difference on the carbon contents, on the carbon stocks in reduced tillage. Still, because initially we only had this information, there's some studies that say that the larger carbon content in the topsoil can lead to increased greenhouse gas emissions. And therefore, we started measuring nitrous oxide emissions. And this is a trial we are running where we not only measure nitrous oxide and CO2, but also we installed rainout shelters to test for the effect of climate or reduced rainfall because of climate change. So then here, we started measuring in February 23, and these are our measurements in both systems, conventional and reduced tillage. So here, this is just CO2, which not so clear. I mean, conventional being a bit larger than in the reduced. And and here we have split it for the two crop seasons. Initially, we had wheat and then barley. And it is on the conventional tillage, the CO2 emissions are a bit higher, with no clear difference between the two rainfall patterns, basically. But when we look at nitrous oxide, I think here is where we see this large difference. So in the reduced tillage, the emissions and the reduced rainfall are much higher than in the conventional. So the increased carbon in the topsoil in the reduced tillage has the potential to increase N2O emissions when under dry conditions, not when the soils are very wet, which means that even if this is not having a great effect in terms of increasing the total stocks of carbon, so the total carbon that is captured in the soil, just the difference in the topsoil plays a role on how much or how high the nitrous oxide emissions are. So the trade-offs here are not very positive. And this study is part of a larger project, which is called True Soil. that we are 13 partners from different parts of the world, and we are all looking at management practices that aim at increasing carbon in the soil or theoretically aim at increasing carbon in the soil. And also we try to check the trade-offs between greenhouse gases. And we have set these rainout shelters, exactly the same system in 4 countries, and a little bit slightly modified in 3 more. And in some others, it was not possible, of course. But yeah, we are trying to compile the data from different parts of the world. And this is very super preliminary data. But just that the management practices we look at are different because they are practices that are relevant for the sites in each of the countries. So in Finland, it's monocropping versus multicropping. We are doing the conventional versus reduced tillage. In Norway, they check a hill where the erosion leads the accumulation of carbon in the lower part of the soil. And then in Portugal, they are looking at biochar. And there's, a couple more. The emissions in Germany, this is total emissions until the moment where we requested data. This is why the emissions in Germany are much higher, because we started measuring earlier. So this is pretty unfair way to compare. This is I just wanted to show that we are looking at these management practices and that we will then compare, of course, in a yield-related way, for example. And finally, in terms of more mechanistic understanding of the process, we are running another project also in Göttingen, where we have installed flux tower and chambers in parallel for nitrous oxide. So far, when I talked about ecovariance, we were only talking about CO2 and methane, because the concentration of these gases in the atmosphere is a bit larger, and the fluxes are a bit larger, and therefore they are easier to measure. And N2O, the fluxes, if you know a bit about this gas, the fluxes happen following fertilization or management practice and are super sporadic. Sometimes the crop takes a lot of nitrogen and then there's not large emissions, but then suddenly it rains and then you get a peak. It's super sporadic. So the mechanisms, so the microbial mechanisms that produce the gases are clear, but in which conditions and how the drivers are not so clear. So then we have a more comprehensive approach where we measure the gases continuously with eddy covariance and chamber, look at management practice, climatic conditions, a lot of variables in the soil, and then look at the mechanisms in detail with isotopes and with metagenomics to get into all the microorganisms, all together in parallel. So running from the large scale of an eddy covariance to the microbial communities from the metagenomics. And we still, data are still very preliminary, but we have static chambers, these 8 points that run continuously, the flux tower, and also because the N2O fluxes are not only super variable and temporally, they're also very variable spatially. We also have made, I think, 5 so far campaigns of 100 points within this tower footprint to understand what's the variability on the soil, and they come of the emissions. Also, they come with soil analysis in some like in 30-something points to try to see the trade-offs. And these are data from 2 of the sampling campaigns where we measure with the chamber over 100 points. And we can see here that the fluxes are super variable within a theoretically homogeneous field. So that even when you would walk around and you look at it, it looks like the field is just, there's no plots, there's just an homogeneous 10-hectare field. And the emissions are still very variable. So this is something that is quite interesting. And we have the temporal analysis of the fluxes. The first year, we had sugar beet, which was fertilized with 60 kilos of nitrogen per hectare. And these are just the data from the sugar beet, and the emissions are very small, but there's peaks that happening not always related to the emissions. This was happening following some rainfall events. So when you fertilize, we make sure that we go with the chambers often enough. When it rains, you try to follow the emissions, but it is quite challenging because the emissions are just very, very sporadic. But we hope that with this super thorough analysis of the temporal variability plus spatial variability, we would get more understanding on how these management practices are affecting the emissions and how can we then help to mitigate these emissions. Yeah, so this is related to the increase of water and temperature. And yeah, here we are looking also at soil variables so that we can also get an overview about the drivers. And finally, this is just an overview of the activities of our group, the environment modeling group. So we are quantifying and trying to get mechanistic understanding of greenhouse gas and energy fluxes between ecosystem and the atmosphere, and to develop mitigation strategies. We work mostly in the field. So we do field experiments, the type of things that we've been talking about. But we also have some laboratory or incubations in the greenhouse to try to get more mechanistic understanding. And we are right now starting to work with different models to be able to upscale from what I've been explaining here, which are data from single sites that give information very specific for a particular site, but are not valid enough for larger recommendations on management practices, to be able to upscale our results into larger areas. So just, as a general overview, agricultural soils have the potential to store large amounts of carbon, but they also emit greenhouse gases to the atmosphere. There's management practices that accumulate and return carbon in the soil while reducing the emissions. So this would be a way to contribute to climate change mitigations. But it's important to measure under specific conditions, under specific management practices, to be able to know that indeed the proposed management practices have the effect that we are really aiming at, such as the example of the reduced tillage that has been given as the example of something we should do, and in these particular conditions doesn't seem to be working very well. And yeah, this is it from my side. This is our flux tower in Göttingen. And yeah, thanks a lot for your attention and open for questions.